So today, I'm very happy to present Joe Kirschvink. He's going to be talking to us today about human motion perception and how it utilizes the geomagnetic field as a compass. It's the subtitle of Full Vector Magnetic Sensory System based on biogenic magnetite. So with that, I'm going to stop sharing, let Joe share, and take it away, Joe.
Good morning, everyone. Let me see if I can get the right share here. Uh, Good morning, good evening, good midnight, depending on where you are. Happy to be here and thank you for the invitation. Uh, Today, I'd like to give you an update of some of the work we've been doing over the last 10 years on the geomagnetic sensory system in humans. As many of you know, I've been working on the hypothesis that animal magnetoreception is based on biological crystals of magnetite, similar to what we know and love in the paleomagnetic community as the magnetotactic bacterial magnetosome chains. Uh, the same system for making magnetite appears to be present in eukaryotes, all the way from protists that use the magnetic field, just like magnetic bacteria, to animal navigation. And there are a couple competing hypotheses for about how the magnetic sense operates in animals. And the thing to put this in perspective for anybody with a neurobiological hat here is that in order to give a whale a magnetic compass would require the equivalent of a handful of magnetic bacterial cells. That's all you would need to produce a compass to allow an animal to navigate. If you actually look at the behavioral data from animals, though, it turns out the magnetic sense is a very, very highly evolved sensory system. It's not just a compass, it's also using fluctuations in the magnetic field as navigational cues, the geomagnetic map, so forth. And the way that you would do that neurologically is not by having a handful of cells like you would need for a compass, but uh, thousands to millions of structures, all giving information to the brain, whereas the brain processes it and extracts important information. And you'd say, well, how important is navigational map? If you're a migrating bird leaving Scandinavia, going for southern Africa, and you're flying high at night, and you encounter a magnetic storm or the magnetic field goes crazy, you could be deviating from your path by thousands of miles. So, and if you don't get to your nesting grounds, you die. So, indeed, there is a strong selection pressure for this. So, what I will do today is to argue that the evolutionary product of that sensory system is also present in humans. And uh, I'll review briefly work we did about published in 2019, showing that humans do have this magnetic sense, and that's most likely based on magnetite. Uh, and then delve into a couple of the other aspects. In the 2019 paper, we demonstrated it was a compass sense, and it was a polar compass. In other words, it could tell uh, down into the north from up into the south. The polarity of the field was resolved. And that's actually important for resolving what the biophysical mechanism was. But we had no evidence at that time that it was actually being used or that the other system, the anomaly system, the intensity map system was used. So anyway, what I'll do today is review briefly the eukaryote story for biogenic magnetite, in a very simple one, and then launch into this and hopefully stay within the time limits for the MagNetZ. So again, to acknowledge funding support, Human Frontier Science Program, NASA, DARPA. More recently, the Foundational Questions Institute supported the work in Australia. And the actual topic that I'm talking about, largely led by Laura Christ, who's a postdoctoral fellow at Caltech, Felicity Meekins, who's a professor of linguistics at the University of Queensland in Australia, and a bunch of students and supporters and collaborators like Shin Shimojo, and Atsuko Kobashi and others on this. So going back to 1961, my mentor as an undergraduate was Heinz Longson. Now, I wasn't there in 61. I joined Caltech as an undergraduate in 71. But 10 years prior to that, Lowenstein had discovered magnetite biomineralization in molluscan teeth, chitin teeth, I think everybody's familiar with. Uh, and he did that actually by noticing that uplifted coral atolls, particularly in places like Palau and Bermuda, the magnitude of these nips increased the further away from wave action you got. Hence, it wasn't wave cutting these, it was another thing cutting it. And he actually looked at it and discovered that mollusks in particular were etching into the rock. And these are, in fact, chitins, polyplacophorus mollusks, who were able to eat into the carbonate rock and feed on the endolithic algae growing in them. And Heinz noticed that there were scrape marks, classic gradular marks. But there was a puzzle. The German text at the time said that the teeth of mollusks were made of a protein called chitin. And Heinz knew that the chitin was like in your fingernails, was too soft to really eat away the carbonate. And so he said that it must be mineralized, and it led him to actually look at these critters. And eventually, through a very long series of X-ray diffraction, realized that it was the mineral magnetite. And that led to this simplest biomagnetic experiment you can imagine, taking the radular tooth organ out and just seeing if it sticks to a magnet, and yeah, you can drag it around. Clear example of biomineralization. He hooked me on that as an undergraduate, as a research project. And the question is, how much of this stuff is thrown out into the environment? Could magnetite from the chitin teeth contribute to the mystery back in the late 70s? How did magnetite get to deep sea sediments? A number of paleomagnetists had worked in that. Nobody had a good answer. Biological precipitation was a possibility. We now know it's mostly the magnetotactic bacteria magnetizing the marine sediments, at least with the fine-grained single-domain components. But we now know that actually, another whole story, uh, shallow water carbonate muds in lagoonal facies, certainly of Palau, are magnetized by the chitin teeth. The coercivity and fork diagrams from the mud match very nicely the eroded fragments of these teeth that you get in the fecal pellets of the animals. Anyway, that's another study. Uh, Chitins mineralize the material. The actual magnetite crystals are about, in this particular species, cryptochitin, about 1/10 of a micron crystals embedded in a collagen matrix on the surface. And that was fun studies from the 1970s. So let's then delve into the human system. And what I want to do today is talk about the COMPASS system, Wang-et-al review, and then new data on the magnetointensity response, and new data that actually the reference magnetic frame is not just a vestigial feature. We now have evidence that it's actively running in the background of your brain as an orientation process. And we have a new experiment largely developed by Laura Christ, where you give a visual illusion in a darkened room inside our coil setup with a random thing called a random dot kinematogram. And as the dots are flashing on the screen, we subtly rotate the magnetic field left or right. We can deflect the apparent motion of those dots, which means when you're just sitting there, your brain actually has this process running that uses the magnetic field as a reference. It's there and it's real. It's fun. So anyway, I'll start with the human compass. This was the paper republished by the Society for Neuroscience in their online journal eNeuro. 50 gigabytes of raw data went into it, whatever. We know all of these organisms have a magnetic sense, bacteria to dolphins and whales. Humans were and remain a bit of a mystery. So we built a magnetic chamber with completely isolated conditions, merit 4-coil designs, double wrap coils for controls and blinding steps. First off, discovery was that the alpha wave pattern in the brain, which is about a 10 hertz background hum that the brain gives when you're just sitting and resting, doing yoga or whatever. If you put electrodes in the brain, you'll hear a very low hum. The amplitude of the alpha wave actually corresponds to sensory input. When you're in the dark and meditating, it's kind of like the clock system of the brain. The different parts of the brain fall into phase and rest when things are nice. If there's a sensory disruption, a loud event, shock, something, parts of the brain that are needed to see whether this is a bad thing or a good thing drop out and the hum goes away. So the amplitude decreases. After a few seconds, it increases again. And what we discovered was that a rotating magnetic field, rotating at 90 degrees in a tenth of a second, produced a drop in that amplitude. And you can actually characterize it. Neurobiologists do this all the time. We had 64 electrodes in the brain, and we can actually see drops that were rather significant that went through that. So, and you can actually see that. And as you argue, it's most likely magnetite. And so here's the description of the setup for a cage, shielding radio waves. We'll get into that. The merit coils, we could generate about half a cubic meter of space around the subject's head, uniform to better than 0.2%. So a very clean magnetic stimulus. Isaac Hilburn was essential in getting that programmed and up and running properly. So brief review of magnetic reception. Three major possibilities. Electrical induction, perhaps works in sharks. Sharks can detect a few nanovolts per meter. It's like a battery across the Atlantic. And so as they're swimming in the geomagnetic field, they ought to be able to measure their induced components. That's one possible mechanism. Notice, though, an induction compass does not respond to a static magnetic field. And 25 years ago, Mike Walker and I actually did an experiment putting little magnets in skates, just fixing them, and it disturbed their magnetic sense, which means that it's probably not right, but we'll get to that into a bit. The second major idea is a quantum mechanical effect. If you have free radicals in a enzyme system, say, such as cryptochrome, where a photon activates a molecule, you go from a singlet to a triplet. The triplet will have a magnetic effect on it, and if you average over billions of these things in the eye, you might be able to extract a compass level sense. Now, the interesting thing about that, it depends on the Hamiltonian of the energy matrix, the Hamiltonian of the system, and that goes as B squared. So a quantum compass can't detect B versus minus B fundamentally. So if you can demonstrate your organism detects north down from south up, you know it's not a quantum compass. And the other option, which of course is my favorite because I proposed it originally, is that chains of magnetite, similar to the chitin teeth, might be involved in that. And there is direct evidence for these structures in animal tissues. These are magnetosome chains that we pulled out of salmon olfactory tissue way back in 1988. And of course, the human brain magnetite, the Kobayashi et al., with beautiful single main structures in the brain. So we know that's one. So this is the EEG experiment, 64 leads on the subject. And we can actually do tests of these three mechanisms, which we did. The induction depends only on the moving, not the static components. So if we put a static upward component and toggle the magnetic field this way and compare it with the static down toggling, we can detect. And so here's the up directions versus the down directions. And again, this is the power level. A drop in alpha power after the stimulus would cause like this. So here you see this drop. It's in the down counterclockwise, not in the up counterclockwise. Clear response. Uh, and that kind of means it's not an electrical current. And so it's not that. So a quantum compass can't detect the polarity of the field. This is the second hypothesis. If we go up into the south versus down into the north, this is downwards B versus minus B. And this one, again, let me run back and show that again. The system here, again, uh, down into the north, we get a very pronounced drop in the counterclockwise and not the clockwise. See, here's the whole part of the brain, something's wrong. You shift the magnetic field, the brain's panicking. It's the first time in that person's life, or these people's life, where the magnetic field shifts and your head hasn't moved. And so that's a sensory disconnect based on your training, and the alpha waves go crazy. And it means it's not a quantum compass. We did this over and over again because that was the main competing mechanism, and chance probability of getting that is less than one chance in 100, very clearly. So anyway, first order conclusion, it's not electrical induction, and it's not a quantum effect. The only viable mechanism is magnetite. And however, we didn't show in that 2019 study that the brain actually used it. So let's go back and say, okay, we have a compass. Is there another anomaly sense? And we were thinking that, how do you do this? And what led us to think about, well, gee, human ancestors were obviously hunter-gatherers, and there are human societies now that maintain their ability to navigate with less sedentary lifestyles in the cities. And I came across work by Felicity Meakin's group at the University of Queensland studying indigenous languages in Australia. And interestingly enough, many of the indigenous Australian languages do not use egocentric terms. They'll say, for example, the Vegemite is on the north shelf, or there's a snake southeast of you 20 meters, run to the northwest. And if you don't know your orientation in the world, you can't communicate, and it could be dangerous. And so we said, well, gee, maybe they're using navigational abilities as well. And so Felicity, it turns out, had been studying the Gurindji language. She actually wrote their dictionary. And this is just the cover for my talk showing former Prime Minister of Australia, Gulf Whitlam, officially returning some of the Gurindji homelands to Vincent Lagunari, who was the leader of the rebellion in 1996, saying, hey, this is our land. Why are we being slaves on these plantations? They rebelled, and the plantation owner said, you're right. This was originally your land. And they officially gave it back, and then the government recognized that a few years later. Many language groups in Australia, the ones closer to the interior, have more and more geographic components. So Gurindji is kind of a mix. Some language uses river drainage as well as just the cardinal directions. And our test site was, we were going to do this in the spring of 2020. In fact, we had arranged all sorts of things for the Gurindji elders to visit Caltech in March of 2020, when the COVID pandemic shut everything down, and I fled to Tokyo for a year, and all of this had to be put on hold. Anyway, while Itsuko and I were hiding in Tokyo for the pandemic, it was clear we couldn't get to the Gurindji homelands, and even our collaborators in Queensland could not get to the Northern Territory. The borders were closed. And so what we did was we said, well, is there a simple test we could do for this anomaly detection that we could do in our home? So in our table at home, we said, well, what about a shell game? And Atsuko and I basically debugged and developed this thing where we have 4 opaque cups, and we put a hidden magnet under one of them and devised a clever way of rolling dice to randomize them. There's the one die you can see here. And I could make a little kit and we can mail things. Mail was working. So we made a bunch of these kits, calibrated the magnets. And the idea is that do it double blind. Takes 2 investigators in a room where experimenter 1 rolls a dice and hides the magnet under one cup. She then leaves the room. Second experimenter comes in, not seeing the first, rolls the dice twice and swapping them, A to C, B to C, or whatever, and a pattern of dice swaps, doing it twice so that really nobody knows where the magnet is. And then the participant comes in with the first experimenter, and it's a 1 over 4 to the nth binomial distribution, very simple statistics and easy to run. Now, to make sure it was working, we used the rare earth magnets disguised as kind of like Oreo cookies, and we balanced it with some copper and brass washers to within the same 1/10 of a gram, the same mass. So it was easy to double blind these things and to make them nice. So we mailed it to Felicity, and this is kind of the setup initially. And a simple wicker table and bringing subjects in one at a time, far away from magnetic noise. And here's the setup there. Typical subject making the choice. Now, the results were kind of wild. So most of the subjects basically did random chance, shown by the green here. This is the number of guesses versus the number of correct guesses. And when you start getting 7 in a row correct, like this one individual did here, and then 3 guesses, the chance of that is pretty small. 3 of the original subjects did that on the first setup. Those 3 were brought back, and some of the ones did much better even on the second one. Another subject came back and did 9 out of 10 correct. On the grid, we also later next year we did this on Caltech participants, and it's clear that Gorenji had this outlier of people who did it extraordinarily well. And again, one of the Gorenji who did extremely well came back a few days later. And probability of an individual doing that is less than 1 chance in a million. Okay, strange hidden magnets on a table. Humans actually have this ability. Actually, it's not just the Gorenji. Even the Caltech participants were significantly 1 chance in 20 deviant from what you would expect by random chance. So this is a hidden sensory modality in humans expressed more intensively by people whose ancestors actively maintain this ability to put. So thinking about this, and of course being at Caltech, we have access to some really good electrical engineering students, and I snared a couple of them into designing an electronic version of the shell game where you had push buttons and a big magnet that would generate the same dipole moment of the other magnets and would do it much more quickly. As Dylan Pearson, for example, actually came to us to Australia and was debugging this even at the last minute when things opened up and we were able to do it. The push button device looks like that. And this is the mounted thing on the table. And in the field, actually, with push buttons, and then we actually filmed it with a GoPro camera in the ceiling. And here's the button thing. I'll kind of show how it looks. I don't know if you can hear me. Uh, so anyway, you can see the magnetic anomaly is the one that's lit, battery powered, and just testing it. You can see the magnet being deflected here. And so anyway, it goes into a game mode where you push 2 buttons at once, and then It records the data digitally and so forth. So it's fun, and the results are astounding. It's much quicker. Again, about 10% of the Geringi displayed hidden runs. I mean, again, it's not absolutely conscious aware. It's not at the forefront of consciousness. But when asked to guess, you get strings of individual runs within these strings where they do remarkably well. And again, we threw every statistical trick at it. Uh, False discovery rate corrections for the number of individuals, uh, Monte Carlo simulations followed by the false discovery rate. And again, as a group, the Geringi do it. Most of them don't, but again, there are strings where people suddenly click on, and one chance in less than 1,000 people doing it. We didn't run that many experiments by any means, and that's after false discovery rate. The Caltech participants were not as good. Again, you would expect 1 in 20 maybe to be significant, but compared to the Geringi, where the reds are really the important ones, no. So bottom conclusion, and here's the false discovery rate scenario on that. Most of them didn't, but the ones that did were kind of up there. So human neurophysiology contains a functioning intensity reception system, the magnetic map. So what that means, it's a full vector magnetometer. It's doing the direction, it's doing the polarity from the Wang et al. Experiments, and the intensity sense is there. Fun. Okay. Uh, what about, is it vestigial or is it actually being run? So here's the new experiment, largely led by Laura Quist. I'm going to show you a random dot kinetomatogram. The idea is that you have a screen, you have a dot in the middle where this red dot is showing. Here you center on it, and then when it runs, you will see the white dots flickering in and out, and there will be a general impression of motion somewhere. So hopefully you can see general motion that way, general motion this way again, general motion that way. So it's a way of fixing your reference in the middle and seeing what the background is. Now, what we do here is we have another version of our magnetic sense system, which is a 1 meter cube. Subject sits inside, and we can project through the Faraday cage from a monitor on the outside. And we give the subjects a push button so they can sit and click back and forth. Laura was brilliant in doing this experiment, let me tell you. Anyway, we controlled the magnetic field. They did the RDK. And the idea is that if you then rotate the field left or right, then you have shams in between where things don't happen. And this is kind of what you find. If you pre-train it where 60% of the dots are in fact moving initially, and then you give a subtle feedback, a little sound to unexplained one tone or the other, eventually the subjects start choosing more one direction than the other. And there's a bias in which way the directions are going. And you can actually see the histogram saying, well, initially the distribution is kind of random. After that first 100 odd presentations, you do the testing with 0% current motions. The patterns are actually not moving, it's just random around the middle. But the magnetic field is rotating still. And what you see is, again, an even bigger bias where the deflections are going in the opposite direction. And I'll get into this. Now, clear sham, a double blind thing where you do everything except the coils are double wrapped. So the magnetic sham condition, the currents are anti-parallel. Everything's the same, just one TTL switch flips relays, and the magnetic field doesn't change, even though the power supplies and everything are doing the same thing. And again, it goes away. Clearly a magnetic effect. So what does this all mean? Well, imagine now, I want you to, all the people watching this, do something. Imagine you're looking at a scene dead ahead of you. focus on a point on that stain, and then with the magnetic field that way. We then shift the magnetic field, say, to be towards the left. And if your field, so the magnetic field is in where that X is, okay? The view will shift to the right if that's your frame of reference. And the other way around, if you shift to the right, your field of view shifts to the left. So what I want you to do now is to put your finger out in front of you, focus on it, and then turn left and right, kind of like I'm doing here. Okay? Do this. Everybody, move your hand back and forth. And if you focus on your finger, you'll notice that the background is moving in the opposite direction. That's the effect that we see. The world moves in the opposite direction. In the random dot kinematogram, we're seeing a deflection opposite the way the field is going. And so, bottom line, we are using this. It's running as a background process in the human brain. It's a reference cue. So it's an active functional magnetic sense and biophysical puzzle. Um, biophysical puzzle. And this is something that started seriously about 15 years ago in the animal literature when it became clear that the bird biologists were unable to get their magnetic hopping experiments in cages to work if their laboratories were located too close to AM radio stations. The best data eventually came out from Henrik Moritzon's group in Oldenburg, where they had built these magnetically quiet chambers and had been unable to get their experiments to replicate. And the idea is that you put your migratory songbirds in a little funnel with typewriter ribbon and tape paper around it so it records the hopping scratch at night. And they eventually figured out that you need to cancel the radio waves in order to get the birds to go the direction that they wanted to hop, the magnetic north, with all of the controls. If you allowed radio waves into the chamber, you got random orientation. And it was very clear results and very mysterious. And it led to the idea that it was a radio wave cryptochrome effect, where you start with the singlet, you absorb a photon, you're going up into a triplet, and that triplet actually precesses relative to the Lamar frequency of the free electron in the geomagnetic field, which would be a little bit above the megahertz for the dipole moment of the electron and the background geomagnetic field. And lo and behold, they found that the orientation did indeed depend on that. And you can shift the magnetic field and so forth. And it led to a whole bunch of people over the last 15 years systematically figuring out what frequencies did and what didn't. They haven't done threshold because it was so bad. Summary paper a couple of years ago, all the 9 results in birds, inhibition showing in red, orientation in green, narrowing the frequency band where this really happens between about half a megahertz and about 80 megahertz up at this thing. And so disoriented, oriented, so predicted cutoff based on the crypto. It turns out, I think you can also do this with the absorption of magnetite containing cells. The absorption of radio wave depends on the imaginary component of the frequency dependent magnetic susceptibility, the Akai double prime factor in the susceptibility tensor. And that actually for superparamagnetic magnetite drops off at about the same level. So I don't really believe it's a cryptochrome idea. But so we did experiments 10 and 40 megahertz on human subjects. Original experiments are RF loop at the Lamar frequency. Why not? And we introduced a double blinding simple short on the RF. And we used a Helmholtz machine, thing for 1 megahertz. It worked fairly well. And here are the amazing results: 25 subjects. This is now the EEG. You can see the nose and the ears and the pattern around the things. So, stimulus artifact. And then, in the absence of radio waves, bang one minute, absence of off. This is RF off. You get the clear effect. If the radio waves are there, this is what you see. So no radio waves, radio waves. So it means a 1 nanotesla, 1 megahertz, or actually I think this was 10 megahertz, uh, radio wave is influencing neurobiology. That's wild. Okay. We've also done it in the new RDK experiment at 40 megahertz for various regions. And again, the training paradigm for that little shift here, big shift. And then here's the sham antiparallel. If we then introduce the radio wave again, it's like the sham. It blocks it. And that's at 40 megahertz, 10 nanotesla peak field. Now, mind you, 10 nanotesla sounds small, but the energy flux across a cell, this 10 micron cell, is many, many kT. There's plenty of energy if you can transduce it. So I don't think that's an issue. So the biophysical puzzle, the ruling hypothesis that the RF sensitivity arises from singlet triplet conversions, but human compass is polar, not axial, and should not be disrupted by F. Yet it's being disrupted by RF, and it's not the cryptochrome. So there has to be a different mechanism. So that's why I think ferromagnetism. So conclusions on all that, natal languages that are cardinal centric uh, seem to have enhanced abilities. Also, an interesting thing that turns out from Felicity Meekins, our collaborator study on this, forced introduction of English to the children of indigenous speakers inhibits the cardinal sense system. And because of this racial British imperialist imposition of English to these communities, uh, the geocentric systems are minimal. And that might be some reason why it's so hard to get this thing in. It turns out there are no untouched indigenous cultures in Australia left because of this doctrine of forced assimilation. Uh, so anyway, that's the pity from this thing. And I might mention that our study in particular is quite distinct from other studies of indigenous cultures. Most past studies have tried to identify a deficit with respect to other cultures. Here we think the indigenous people of Australia are giving us a key insight into how magnetoreception functions in her human neurophysiology. It may actually be a stepping stone to an eventual goal, trying to train all of us to be aware of our intrinsic magnetic sense. And we hope we can get there. And the random dot kinetomatigrams, we're using it. It's not vestigial. It's one of many orientation systems that are running in the background process of the brain. The brain's a computer, just like, say what? All right, anyway, uh, it's polar in nature and should not be able to be disrupted by RF radiation, but it is. Thousands of people complain, by the way, of electromagnetic hypersensitivity, but the current medical dogma does not recognize this diagnosis. And if you want to, we can go into a discussion about that, but I think it actually might be a real malady in many people. The two human magnetoreceptor modalities are novel and they have clear biomedical implications. So I'll leave it at that and open it for questions and comments and so forth. I think I'm on time.
Thanks a lot, Joe, for fascinating talk. I think everyone should just give a virtual round of applause for that. That was
Okay.
really cool. Yeah. And yeah, open the floor to any questions. You can ask questions in the chat or just raise your hand and then can pick you. You can unmute your mic. Just wait.
I see a comment there in the
Hold on a
chat.
second.
uh,
Someone with that hand raised.
well, thank you for the comment, Arroyo. Actually, interesting to note that we really have no idea of what all the magnetite in tissues do. We know human brain tissues make magnetite, but many other tissues as well. Uh, Beef tongue, for example, is highly innervated organ, sensory receptors for taste and touch and temperature. There's also magnetite in beef tongue, and we can actually get clean samples of that. There are cells that we're calling massively magnetic cells. Using scanning squid imaging with the squid sensors over fixed tissues, we can actually see occasional cells that are extremely magnetic. Uh, those might be some of the radio wave sensors. In order to detect the radio waves, you would need cells uh, 10 microns or so in size that uh, would be able to pull the imaginary response, well, the energy absorption from the imaginary component of the susceptibility tensor and actually get into that. So, how many of you have magnetometers that can do rock mag analyses under kind of clean lab conditions? Oh, do I think the topic? Okay, let's see.
So yeah, there's a couple of
Uh,
questions in the chat, one from Nick Jarbo and one from Peter Driscoll.
yeah, okay.
Well,
If radio waves disrupt this magnetic sense, do you have an idea where or what in the modern world would do this? Ah, okay. Yeah, it's within 10 kilometers or so of a typical 50 kilowatt, 1 megahertz AM radio station. So basically, any large city, your magnetic sense is going to be interfered with. Nice thing about the Gurindji village, there's only one local AM station, and we measured the intensity of that, and it was below the threshold. Uh, Airline pilots, it may be a contributing factor to spatial disorientation errors. Do I think it's understudied? Hell yes. We there's an incredible need to understand the distribution of magnetite materials in tissues. There are a few studies, Stu Gilder's group, and among others, who looked intensively in brain tissues. We need more of those studies. We need to be able to find the tissues intact with the cellular uh, things there. There's so much to do. Uh, let's see. Yeah, it's understudied. Edward, hi, greetings from Prague. Well, actually, we maybe I shouldn't reveal this, but in I'm now part time in the uh, Kochi Marine Corps Research Institute in Shikoku, Japan, with Yuji Yamamoto's group and others. Uh, we did we've started a collaboration with the medical school there, particularly with the group studying autism. There is some really weird distribution of autistic children in Kochi, including one elementary and middle school where something like half to a quarter of the students are on the spectrum. Particular school is located a stone's throw from a major uh, radio station in HK. There may be there may be other aspects to this. If it is a human sense, it might be overexpressed in autistic people.
Okay, thanks a lot and best regards.
My pleasure. Uh, uh, Gunther, we don't know. This is why I'm saying, Gunther's question is, where's the location of the magnetite in the neural cells? We only know in one area, and that was the study 25 odd years ago by Mike Walker's group in fish. DL and Walker and others did electrophysiological recordings in juvenile salmon, excuse me, trout, same genus, and were able to find the neurons in the supraophthalmic trunk with electrophysiological recordings that were magnetically sensitive. They move the magnetic field and they can see spikings in those cells. They then injected from the same electrodes a dye that was diffused along the lipid path and radiated out, and they then followed through by histological studies and focused on the olfactory epithelium in the nose. And it turns out some branches of this nerve started there and then brought the information back in. They then went in through this tissue stains, stained for magnetite, located things, and went in with the magnetic force microscope and were able to demonstrate single domain particles. They could magnetize and change the orientation in those cells. They resolved the cellular structure there, 15 to 20 micron size cells that were synaptically connected to those neurons. Inside those cells, there was a vesicle about 5 to 10 microns in diameter that were loaded with magnetite. So that's the clearest image of these receptor cells that we've been able to have. And so, yeah. So, Uriel, do I think disorders of magnet in the brain? Yeah. Dunn's work on human brain tissues has shown very clearly that there's also supraparamagnetic tissues there and material there in the brain. My best guess for what the ultrastructure is of this receptor is a cell loaded with supraparamagnetic crystals. Now, it's either this, because the supraparamagnetic crystals, they peak in the imaginary absorption at about 80 megahertz and drop off, which is the pattern the bird biologists see in their behavioral data. And it kicks in when you start getting the ferromagnetic resonance effects above about 1.1 gigahertz. And so I think the increased supraparamagnetic materials in the brain tissues might be a result. Now, again, I know I published an abstract in 1992. I think it was an AGU meeting where we took a lump of brain tissue, froze it in liquid nitrogen, magnetized it, and then controlled its warming inside a 2G magnetometer. And you could actually see the supraparamagnetic fraction becoming at room temperature, transitioning from stable single domain to supraparamagnetic as it warmed. And you could involve that and get the size range. And it's there, it's active, and yeah, maybe it has something to do with the Alzheimer's disease. Stuff is certainly there.
Hey, well, I think if there are no more questions, we can wrap up this part of the session that is recorded. Give Joe another round of applause.
Thanks.
And yeah, no, I think it was a very cool talk. Yeah, stop the recording just to let people know that the MagNetZ YouTube will be, if you missed the beginning of this talk, you'll be able to catch it on the MagNetZ YouTube. We've got, I think, coming up to about 150 videos on the YouTube now. So if you've missed any of them, be sure to catch up on them there.